In this work, we studied the evolutionary history of most intronless genes and their orthologs in 10 species, including human. Then we compare it with the results of the same analysis for multiexonic genes. We aim to contribute with evidence that can help to determine which theory of single exon genes origin corresponds to intronless genes, the introns early theory or the introns late theory. The evolutionary history of a set of genes can be represented as an even-labeled gene tree, where leaves represent genes. Inner nodes are either speciation or duplication event, and the relation between any two genes is the last common ancestor in the genes tree. The reconciliation of genes trees with a species tree reveals in which branch gene duplications and loss occur. In this work, we show that the reconciliation of the evolutionary history of intronless genes in mouse contribute to the introns early history, and gene ontology analysis highlights that intronless genes have specialized functions. Genome of 11 species were downloaded from the ensemble database. We identified the set of 1,116 intronless genes and 9,017 multiple exon genes. Protein ortho was used for orthology inference. Then we applied modular decomposition algorithm to each orthogroup. This tells us if the set of inferred orthology relations has evolutionary sense. If not, we apply a mean cut heuristic for edit it. The modular decomposition gives us as output the even-labeled genes tree that contains the evolutionary history of the genes. This tree is reconciled with the species tree to obtain gene ancestry and the branch of a species tree where evolutionary events occurred. We deduce how old a gene is by the identification of orthologs in 4 related species. We also identify the intronless genes that are species-specific and intronless genes that were lost in ancestral genomes. Finally, by gene ontology analysis, we identified the functions in which intronless genes are involved. The reconciliation tree summarizes the evolution of intronless genes. For each node of the tree, blue numbers indicate the numbers of genes corresponding to ancestral or existing species. In each branch, green and red numbers indicate gene duplications and gene losses, respectively. At the right of each reconciliation tree, an offset plot shows by columns the number of ortholog groups that are conserved in a complete clade. Note that a higher number of intronless genes are only shared by rat and mouse, since they are not present in any other species. The heat map shows that intronless genes are specialized for Rodentia and Plantae, and the functions of these genes are involved in detection of chemical stimulus and DNA replication-dependent nucleosome assembly, among other functions. The bar chart shows how the most intronless genes orthologs are classified. Note that in all species most of them are also intronless genes, and the proportion of multiple exon genes decreases for species closer to mouse. These results seem to indicate that intronless genes are more recent and specialized than multiple exon genes.